Hello friends, welcome to yet another intriguing edition of Rahul's Advanced Biology. Today I'll be bringing you a very ecstatic topic known as COVID-19 and blood clot formation in the lungs. Before I start, I would like to tell you that the WHO has already declared the SARS-CoV-2 virus-induced disease COVID-19 as a global pandemic. More than 3 million people have been affected by it and more than 3 lakh people have already lost their lives. Scientists globally have been working really meticulously in order to come up with various vaccine regimens and various antiviral drug regimens in order to cope and curb this specific pandemic and this specific SARS-CoV-2 virus. In my previous videos, I've already talked about the virology, pathophysiology, and the drug combinations like azithromycin plus chloroquine, hydroxychloroquine, remdesivir, tocilizumab, and so on in order to curb and curtail this SARS-CoV-2 virus. Now, in this specific lecture, I will be talking about the specific hemodynamic changes induced by the SARS-CoV-2 virus. Now, for the beginners, I would like to tell you that the infection by the SARS-CoV-2 virus, which is a positive-stranded, non-segmented, single-strand RNA virus with a genome size of about 30 kb, infects specifically the type 2 pneumocytes, and the type 1 pneumocytes also can infect the hearts and kidneys via binding to the ACE2 receptor, angiotensin-converting enzyme 2 receptor, which is the type 1 membrane protein and which is expressed by the type 2 pneumocytes, the heart cells, the kidney cells, the endothelium, and various other cell types. And the priming event is the most important event. In many research papers published in the journal Science, it was shown that the ciliated epithelial cells, the goblet cells present in the nostril, they express the TMPRSS2 and ACE2 protein together concomitantly. That is the transmembrane serine protease 2, TMPRSS2. This is a type 2 membrane protein, and which cleaves the spike protein present on the SARS-CoV-2. The spike protein is a trimeric spike protein but has 2 functional domains, S1 and S2. After the priming event takes place, it becomes or it forms 2 functional domains, S1 which is the receptor binding domain which binds to the S2 receptor, and the S2 which is the class 1 membrane protein which causes the membrane fusion and the membrane tethering or membrane anchoring. Then it goes into the cells via the endosomal route. That's why hydroxychloroquine is of primary importance, because it can increase the pH of the lysosomes. It's a lysosomotropic agent, thereby it can prevent the entry or the viral ingress till an extent. And we have also talked about remdesivir, and in my next video I'll be talking about EIDD2801, which is also an ATP analog just like remdesivir, which has an advantage that it can be taken in an oral way, in an oral form. It can be taken orally, whereas remdesivir is required to be taken intravenously. It is required to be administered intravenously only. So you need to be hospitalized in order to take remdesivir, but if EIDD2801 gets the approval, clinical approval, then one can take it even by staying at home. Now coming back to our topic, what happens is after the viral ingress, it can wreak havoc, it can make new virion particles, it can synthesize its main protease and the papain-like protease, it can synthesize and it can cleave the various ORF origin, the open reading frame proteins ORF1A and ORF1B and ORF1AB and so on. Then what happens is it can make the subgenomic RNA and the genomic RNA combination. Then in order to curb this thing, what happens is the scientists have come up with various drugs. So in this very lecture, I'll be talking about what are the ramifications of the SARS-CoV-2 virus as far as the clotting in the lung is concerned. Now what happens is ARDS is induced by COVID-19. The COVID-19 disease causes ARDS, acute respiratory distress syndrome, which I had talked in a little bit of detail in my video of cytokine storm. So I'll just reiterate for you that suppose cytokine storm is happening. The cytokine storm happens because of the immune regulation, because of the immune pathway, the IL-6 upregulation. So the IL-6, specifically the soluble IL-6, binds to the soluble IL-6 receptor. The IL-6 binds to the soluble IL-6 receptor, and then this specific complex can bind to the GP130, which is expressed on most of the cells, and can bind to the GP130 dimer. Then suppose it binds to the endothelium by the trans-signaling mechanism. What happens is it can cause the formation or the upregulation of vascular endothelial growth factor, VEGF in endothelium. It can cause the downregulation of E-cadherin. It can cause the upregulation of MCP1, macrophage chemoattractant protein 1. It can cause the upregulation of IL-8 and IL-6. Thereby, it can increase the vascular permeability, which is one of the hallmarks according, and also it can cause hypotension, which is again another hallmark of acute respiratory distress syndrome. These things can happen. But the ARDS, which is caused primarily by the pneumonitis, the type 2 pneumocyte inflammation, pneumonitis present in the lungs, in COVID-19 differs from the ARDS occurring due to any kind of streptococcal bacterial infection. Now what happens is, in case of, you can say bacterial infection, in case of normal pneumonitis or ARDS, what we see is diffuse alveolar damage, DAD, and type 2 pneumocyte hyperplasia. These two specific clinical observations are not profoundly observed in the ARDS caused by the COVID-19 disease, caused in the COVID-19 disease by the SARS-CoV-2 virus. In case of ARDS caused by SARS-CoV-2 virus, what happens is there is septal injury, and septum is the alveolar wall, as we all know it. It is made up of the epithelial cell, the endothelial cell, and in between the epithelial and endothelial cell, there is interstitium made up of connective tissue, which can be made up of collagen fibers, elastin fiber, reticular fibers, and also alveolar macrophages can reside in it, mast cells can reside in it. So what happens is the junction, the tight junction is connected to in the epithelial tissue. The epithelial cells contain the tight junction, whereas the endothelial cells contain the zona occludens. So the main prevention of any kind of entry of any proteins into the alveoli, into the gaseous surface, is prevented by the tight junction present between the type 1 and type 2 epithelial cells, which are also known as pneumocytes. Now, septal injury is seen, and also the fibrin deposition on the mural and luminal surface of the septum has been observed, which is a blood clot. What has also been observed is the infiltration or penetration of the septum, of specifically the interalveolar septum by neutrophils, causing neutrophil extracellular traps, which I will be covering in my next video, the immunobiological dynamics of NEP, neutrophil extracellular traps, NETs. Then, what also happens is there has been a POSI inflammation, which is found in case of COVID-19 induced or SARS-CoV-2 induced pneumonitis or ARDS. POSI inflammation is little inflammation, which is not that exaggerated, doesn't have that much of an hypersensitivity response when you go for IgG mediated immunofluorescence assay. And for an interesting thing, it has intrigued me a lot, especially that complement pathway terminal products like C5b9, which is the membrane attack complex, C4d, MASP2, which is the membrane mannose binding lectin associated serine protease 2. Lectins are the carbohydrate binding proteins. So mannose binding lectin associated serine protease 2 have been also found to be present in the type 2 pneumocytes, and have been also found to be present along with the spike protein, in conjugation with the spike protein of the SARS-CoV-2 virus. So this also causes or brings in more of an immune response, more of an aggregated immune response, a kind of hypersensitive response, and it can also trigger or cause increase in the vascular permeability along with the trans signaling caused by the IL-6 pathway. So these dynamics and the fibrin deposition onto the luminal and the mural surfaces of the septum really has caused a lot of stir in the medical industry, in the minds of doctors, that people have been administering now clopidogrel, aspirin, and other antiplatelet, anti-clotting agents to the critically ill or severely ill SARS-CoV-2 patients, because there have been reports of them having deposition of clot in the lungs, clot in the arteries, and also suffering from cerebrovascular accidents, right? So that's about it. That's all the conceptual prowess you needed to attain in order to completely comprehend this lecture. If you like this lecture, then kindly hit the like button. If you have any problems, any queries, kindly do not hesitate to post your comments in the comment section below. I'll be replying as soon as possible. In the description box below, I have put the links of various research papers and also of my Facebook page. If you want to contact me personally, you can like my Facebook page and contact me via Messenger to get my prompt reply. And if you have again liked the video, then kindly subscribe to my channel Rahul's Advanced Biology, and do not forget to hit the notification bell so as to be notified whenever my next video comes online. Thanks a lot. See you soon.